And today she'll talk about using R generally and also specific about software that she's written for using R in fMRI analysis.
Thanks. So first of all, I should explain that what I mean by doing fMRI in R is the modeling part of that. I'm not talking about all of the pre-processing and all of the removal of noise and all of the registration parts of that occur. There are packages in R that are available for that you can do that also with packages like FSL and AFNI and shell scripting. You can do it in NiPipe. You can do it in anything you like. But what I'm most interested in is solving problems that we don't know how to solve. And those things, I think, are best done with R. And I'll hopefully make a case that you can do fMRI modeling in R, not just that you can do it, but that, in fact, you might want to do it because you have a question that you would like to answer. So before I talk about fMRI data, first of all, could I see a show of hands? How many of you use fMRI data in your research? Okay, so I don't need to go over, I think, a lot of task-related design or anything like that. People have a pretty solid grasp on that? Is there anyone who, okay, good. All right, that's perfect, because I can skip right to the good stuff then. So I'm not going to tell you about the bold contrast medium. I'm not going to tell you about the fact that everyone knows it's really slow and it's unfortunate. I'm not going to tell you about the fact that there's a whole lot of noise that you have to get rid of. And like I said, best tools to do that are in other packages. I am just going to tell you a little bit about, just a reminder about block designs. So the thing is that the fMRI signal that we measure itself, it doesn't mean anything. It doesn't have an absolute value or meaning associated to that. So the way that we interpret that is by comparing it to something else. So typical experimental design, as you're probably familiar with, is that you create some sort of task. And you look at what's called the contrast, because of statistical reasons. You look at the activation of in one condition versus another. And that is how you interpret what is going on with the fMRI data in the brain. So the simplest kind of design, and the one that I learned first, is a block design. And I was doing an NBAC task. And so this is what I use in every single example that I give. In an NBAC memory task, the basic idea is you're trying to look at what parts of the brain are activated under different memory loads. So zero back is a condition where you're seeing a set of stimuli and you push button when you see an X, right? So you're just attending for some letter, or it could be something else, but I've typically done it with letters. And so if you're looking at the activation for watching the stimuli and pressing a button when you see an X versus some visual fixation condition like seeing a cross on the screen for 30 seconds, what you're measuring then is what voxels in the brain are active at this zero back condition, more significantly so than when there is just a fixation cross. And so what you're getting at is through that subtraction, the idea that there's activity related to seeing that X and paying attention to that is more than just getting a visual stimuli. Okay, so and we can extend this idea. So we have a one-back condition where we're looking to attend to whether the letter we see is the same as the one that preceded it. And we can compare that to baseline. And we can then sort of look at, well, what's related to this memory-related activity now, right? So it's not just response, it's memory. And we can do more than that. We can do a one-back versus a zero-back and isolate it to just the difference between seeing a letter that you're trying to remember versus not. You could ask a question that very few people do. Is there any activity related to one back at all? Is one back greater than zero, right? So what is the mean of the activity related to that? And then I have two back versus one back. I will get back to this task design in the future. But this is typically the way, how many people use FSL here? Okay, so you've all seen the design on the right-hand side as some sort of event-related or block design that you've created. What you do when you type in your explanatory variables, and that's what these are called, into your software package, what you get is a design matrix. This is a very simple design matrix here. Because it's only got the three explanatory variables. So this is a block design representation of these three explanatory variables, EV-STIM1, EV-STIM2, and EV-STIM3 in the three different colors. So you see that we've got 1, 2, 3, and then a rest. And then here you can see this in FSL syntax form, where you've got the three explanatory variables, and you create the means for those by setting your contrasts as such. Okay. So what you're doing once you've set this up is you're running a mixed effects model on your data, largely, all right? And I wanted to say, oops, I think I went past one. So I wanted to just talk a little bit about mixed effects models, because this is critical, and this is what we can also do in R. And so this is the foundation for what I'll show to you in a R markdown document that you can simulate data and work with. So we are concerned with things like fixed effects, the effect of gender, for example, or the effect of treatment or control group, or whether the person has a disease or not. And in this case, that would be something where you know all of the groups. You don't have any groups out there that you have not sampled. This is very much in contrast to the data from one individual person with fMRI data, right? Because you have some set of subjects that you've collected for your study, but that is not every single subject who would be coming for your study. So subjects themselves, the data that is within them are random effects. So you model those as if they come from a probability distribution. And what you're concerned about there is not the fixed effect of person A versus person B, but what is the variance surrounding that? And if you don't account for that variance, you're going to get p-statistics that don't make much sense and are not generalizable beyond the population. So this is why we use mixed effects models in fMRI. So the model that you're running looks basically something like this. And I won't go a lot into math or anything, because I'm going to lose myself and I'll lose all of you. But basically, for each of the n subjects that you have, you model the bold signal y of k, where that k is the subject, by some design matrix times the beta weights that you want to calculate. So all of the things that you're calculating are betas, beta coefficients, essentially. And you're interested in, not probably if you use FSL, those coefficients themselves, but you are interested in the significance of those coefficients. So you are modeling it as some design matrix and the weights of each of those things, and then some error, right? So, and this is described here. What have I left out of this? What else do you model, typically? Throw something out there. What's in your design matrix? It was a very simple design matrix. Sorry? Motion parameters. Did you say? Yeah? Exactly. Motion parameters. What else might you put in there? Reaction time? If you're interested in something related to the reaction time specifically, yes. If you're interested in the age, yes. So covariates, you're including covariates, yeah. I'm thinking about all the other nuisance parameters that you might include in there, right? So for those people, who does resting state fMRI? Okay, so what kinds of things do you think about with resting state? Physiological noise, right? So there's no reason for a task design that you have to omit those things, right? And some people do. You can think about bandpass filtering, for example, as a project where you include the regressors that represent the different frequencies, and you can put those in there too. But these are all things that go into this design matrix, right? And I think that some of these get shoved under the rug a little bit when you're working with fMRI software, that you might not be aware that they're all in there because they're occurring in different places, right? So, but you have to kind of understand what you're actually doing if you're going to take it out and take control of it and do it in R. So in matrix form, this is what you have. This is your nice little fMRI time course, and these are your values that you've got. And here you have your matrix with all of the nuisance regressors and your design matrix. But I'm just going to, right now, for simplification for this tutorial, I'm just talking about a very simple design matrix just with your explanatory variables. These coefficients, these beta coefficients, are what you're going to calculate, and this is what you care about, these and their significance. Okay, and then this error is error variance. Who looks at like how much error variance you have left over? Has anyone ever done that? A few people. Yeah? What kind of numbers do you see typically? Like how much of your task data is typically accounted for by your design matrix?
Oh, not very much.
Not very much, yeah, exactly. It's in contrast to a lot of like behavioral types of designs where you think about modeling behavior, and if you only explain 10% of the variance in what you're trying to explain, that would be ridiculous, right? Psychologists would have a fit, they would reject it from the journals, all would be over, right? But this is normal for fMRI design, that you explain only a very small amount of the variance. But we just kind of do things differently in fMRI. We often don't look at that. Some tools make it easier to do so than others. Now, typically, you run that first level model for each subject, whether you're using SPM or FSL or AFNI. That's kind of the way it's been done. And this has usually been done for computational reasons. The way that studies usually work is that you get subjects one at a time, you have your design matrix, you make sure that everything runs, you set this up, and you run this for each subject as you come in, right? Then, once you've done all your first level analyses, you put that together in a higher level model. And then you run your group statistics with a new design matrix that represents your group contrasts and covariates and things like that that you care about, right? Okay, so one of the reasons that that's done is practicality. If you had to rerun to see whether you had something significant, you know, typically, I'm not saying this in the guise of like that people are doing p-hacking or anything like that, but typically you set aside some point where you need to generate some preliminary results for your study, and you decide that you've got this many people and you're going to do some preliminary analysis and see what happens with some hypothesis that you have. If every time you did that, you had to rerun everything, if every time you got a new subject you had to rerun your group analyses, and the single subject analyses, that would take quite a lot of time, because I think people have noticed that sometimes running the single subject analyses take quite a bit of time. So it's more efficient to do each subject separately and then group those. So statistics have been derived to do that, and different methods for dealing with that estimation. But essentially what you're doing is just a mixed effects model. You're just doing it in 2 levels. So, and I won't go into details on that, but there's a tutorial that this is based on that does explain that in some more detail. But essentially, it's the reason that you don't put this into R typically from the start has been computational. It's been because the limitations on memory and on compute for a single machine have been small, and they're not anymore. So I'm making an argument that we should perhaps reinvestigate some of the assumptions that have led to this. Not that everything is bad, but that we might be able to do a little bit more. Okay, so I have a demo here for fMRI in R, which if you are interested in following along, you can go to this GitHub site and pull that repository, or you can just watch. You would need, on your laptop, you would need R, which I think you have from last week, that most people have been using that. And you would need the NLME, which is a modeling package, and the fMRI package, which is an fMRI package. So,
If you're trying to type that in, that's an IM, fMRI in R, not fMRI LNR.
yes, it's fMRI in R. Thank you. So I'll just give you a minute if you would like to do that. I'll leave that up. Has everyone copied that who wants to? Okay. So that repository doesn't have very much. It has a cut down version of our paper, which is a tutorial on modeling fMRI data using a general linear model. And so this is an R Markdown document. You can knit that and make it in HTML. The HTML is also there if for some reason things don't install. Oh, okay. I'll get that. Okay. Try to make this a little bit bigger so you can see better. There's nothing really like dynamic about this. I'm just going to walk through and explain what these parts mean so you can follow along and see it. But the key thing is that if you have that package, then you have the code to do this. So what this is a simulation of fMRI data, right? So just at a single voxel for some 10 people. And we simulate the data using a mixed effects model so you can see what it is and how it's written in R. And then we analyze the data using the mixed effects model, right? So all of the parts of this should come clear, all of the parts that we're modeling if you walk through this. So the first thing that we do is just load these packages. NLME is one of the two main mixed effects modeling packages in R. The other is LMER. Both of them are very popular. Some people love one and not the other. I just happen to know NLME a little bit better, having learned it first, so that is what I'm using. But there's no particular religion associated with that. fMRI is one of the many packages that exists to help you to do fMRI analysis in R. So it's got a variety of functions, but one thing that we like about this one is that it helps you to set up the timings for your fMRI experiment, and it helps you to do the convolution with the hemodynamic response function, and it has support for a few different types of functions. So we're definitely going to need that because those explanatory variables get convolved, and you'll need to do that. That's usually something your package would do for you. So let's assume that we have 10 subjects, and then let's choose our number of TRs. And so we just choose that randomly to be 180. We have an example task with 4 conditions, and I'll assume there's 1 task run per subject. And by the way, I should say, please interrupt me if I'm going too fast or if this is not clear, if you have questions. If everyone's quiet, I'll just assume that it's making sense. So there's a baseline explanatory variable here, which is your fixation cross condition. And then there's a 0 back, a 1 back, and a 2 back. And I've just given the names here in the text, so EV1 through 3, so that you can follow it. So the idea here is that we're going to do a task just like the one that I showed before, where we have a 0 back, a 1 back, and a 2 back, alternating with a fixation cross for some block period of time. So, and then we'll do 15 volumes, 30 seconds for each condition. And we repeat this sequence 3 times. So I'm not randomizing here, right? You would normally randomize. You would do a lot of things to organize these things a little bit better, and you would go for longer so that you have more statistical power, but this is just a small simulation to keep it manageable, all right? So note that. And how familiar are people with R? Like, how many people feel comfortable with R? Okay, so about half. So I'll go a little slow and just explain what these are. So bear with me. If you're an R expert, this will be pretty simple, but otherwise I think it might be impossible to follow. So here, what we do with this thing is we'll set up an empty block where we repeat 0 15 times. So we're just setting the value for an empty block to be 0. So that's all that is doing, is creating a vector of length 15 with zeros in it. Same thing for task, creating a vector of length 15 with 1s in it. So now what we do to create our stimuli is we take together some sequence of empty blocks and task blocks and then repeat that whole series some number of times. So this is basically how we are composing the stimuli for EV1, for EV2, and EV3. So here it's empty block, task block, empty, 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 task, empty, 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 task. So it's very much following along what the picture that I showed you before was. So, and then we want to plot this. So remember that T is our number of TRs. And so we're just going to, this is just a simple plotting mechanism here. So we're going to put the volume number along the x-axis and make the default stim1 black, stim2 red, stim3 blue, and then just put a little legend. So if you do that, you get your little boxcar plot, okay? So while it's really easy to see that we've got all of our EVs correctly, perhaps, and we know when we're supposed to show something and not. We haven't convolved this at all. So this is where we go to the syntax of the fMRI package. And the fMRI package, because it's fMRI language, has its own way of just defining things. And so typically you talk about onsets and durations of events. So that's all this does here is okay. So here, this is just setting your base onsets to be the number of TRs, and then you give your EV1, 2, 3, setting that these are your onsets, and we offset them by a certain amount, and we give them the durations. So presumably this all works out to be the same timings as what I just did with hacked out R, essentially. This is just some convenience functions for people who are used to dealing with fMRI to create the timings. And then similarly, we can plot these to visualize these. So, and we've got that, right? So this is a hemodynamic response function. I believe it's the default one, canonical. There are different ones where I was surprised when I saw this that their drop-off is so high, relatively speaking, right? So I've not seen that typically, but you should, I don't know if you know what your hemodynamic response function looks like, but in the software that you use, but that's something that you can choose. Or many software packages have support for a set of basis functions that you can use to match whatever it actually is, or at least come close to that. Okay, so now that we have our design matrix, let's simulate some data for some actual individuals. So I'm going to skip through, basically, this is just a kind of a recap of the math that I was describing before. We have, for each subject, we know that their bold signal is the product of the design matrix and these beta weights, plus some error. So to simulate a subject data, what we can do is we can pretend that we know what those beta weights are. We're going to simulate those beta weights, and we're going to use some error that will have some random error, right? Error is usually random. And then we can back calculate what the y's should be. So here, I've just made up some numbers. And if you are playing along with the R Markdown document, by the way, the nice thing is you can change any of these and see what happens, and rerun. So we just are choosing some betas. So for subject 1, what we get is, this is our beta vector. For all the subjects, actually, because we're fixing these. So then we have to decide what kind of standard deviation of the error we want, right? So and that's the residuals within subject variance. So we'll just set that arbitrarily to 0.05 or 0.5. So now, key here is that we can simulate the time series for a single voxel. So this is equal to the intercept, which is your beta 0. That's the fixed beta, times the explanatory variable 1 times beta 1, explanatory variable 2 times beta 2, 3 times beta 3, plus R norm is basically just a random number generator that generates numbers between of standard deviation as within dot standard deviation set, so that's 0.5, and a mean of 0. So this is generating some very simple fMRI data. And we can take a look at that. And we can do the same for subject 2 and for subject 3. Right, and we can plot these. So this doesn't look terribly much like what you see when you see subjects. And if you look at say the voxel that most accurately, most significantly matches your design matrix, what's missing here? What's different about these subjects and real data? When you look at your results, they typically look a lot noisier, right? A lot noisier. When we generated this, we said that every subject is going to have the same beta. Right? That's not that's not the case, the same response. So you're likely to see, you know, an NBAC1 and have maybe a different response compared to baseline than somebody else in the room, right? So, and that would mean having a different beta weight for that particular condition. So what we're not accounting for here is the between subject variance. All subjects are behaving exactly the same except for that within subject random error, which was in fact the same too for each person, right? So that's why these look so relatively beautiful. There's a pattern to each of these that they're following along within some error, but that's not what fMRI data ever looks like, right? So we need to look at what we do for group analysis. So, and I'll skip through the math part of it too, so you can just look at the R. And here we just set a seed so that we can repeat this a few times and get the same kind of results. But let us say now, let's specify that each person's beta estimate should vary within a certain range. And that's what these are here, is we set that we're going to have for a standard deviation for each of these beta weights, so that between subjects we're going to get different betas, right? So this is, when you do a first level analysis, I know FSL the best, so I'm going to use that as the example. But when you do an FSL analysis, you have the means, the copes, and you have the var copes, right? The var copes are the variance around the S parameter estimates. And you pass those up to the second level, to the mixed effects level, so that it can better estimate what the statistics should be at the group level. Okay? And that's exactly what you're seeing here. This variance is what you're adding to these beta estimates, which are your parameter estimates, essentially. So, so what we can do here is we'll set that and look at the actual random effects. Okay, so I just printed out some things. Great. Okay, so we can see that we've recovered some variability in that. So now what we'll want to do, the goal here is to get to how do we take all the simulated data, put this in a format that R can deal with, and run a mixed effects model on it. And we should be able to, when we do that, recover those beta weights that we put in. And they should be statistically significant too. So here, what we do is we're just going to write a little function to generate, again, this is sort of just of interest in R. We just write a little function here which will generate the subject voxel data. So part of this looks exactly like the previous part of it. But now what we have to do is we have generated the between subject variability, so we're going to add these in here as well. So this is where we have our explanatory variables times the between subject variability that we generated plus our residual error. So this is generating now a more realistic subject bold response in response to this experimental paradigm. And then this just takes it and says, let's, for n subjects, let's just apply it to all of the, apply this function to 1 through n, and that will give us a data frame of values. Now, we're going to need to convert this into a data frame of the format that R expects for NLME modeling. So this is going to need to look like this. How many people have used NLME or something like that, long data format in R? Okay, for you guys it will be familiar, but I'm, again, losing people. So I'm going to go a little bit slower and hopefully understand. There are two data formats that you typically use for statistics. There's long and there's wide for longitudinal data. So long data format is very simply that if you have multiple identifiers or time points, or, well, not identifiers, I guess, but time points or other things like that, you put all of the things that are associated with each time point with, like, you know, here, my variable of interest, time 1, variable of interest time 2, and those each become columns. That's in the wide format. In long format, what you do is you stack everything so that you have all of the ID numbers, and if you have time points, for example, you would just stack them, and you would have all of the data variables that you cared about for several different time points. And so that's how you would do that. But basically, the idea of a long format is that it ends up being narrow and tall. So here we stack all of the ID number is the same for all of the TRs, where we don't actually have the TR in this particular data format. Here, we have the bold signal here that we've created, and then we have the explanatory variables that we've generated. And again, no nuisance data. So we need to get it to that format. And this is just some machinations for how we do that. We take all of that subject data that we created, we use unlist to break it out of the list format. This is just mechanics. We create a variable that is the ID number, and that has to be a factor so that you don't use it as a mathematical term, especially when it's something as simple as 1 through n, it's numerical. We need to replicate the explanatory variable vectors so that we can stack them appropriately in this data frame. And we create the data frame and call it voxeldat. And you'll see this when we talk about the NeuroPoint software. So it's going to be exactly the same format. So here, this is your voxel data, just for one voxel. But since we typically do a lot of modeling on a voxel-wise basis, this will get you quite far. Okay, now that we've created this data using some expectation that it would follow the assumptions of a mixed effects model, we can go forth and actually use that mixed effects model to model it. So here, this is what the command looks like, LME. We specify here that our data comes from voxeldat, so that's where we get all of our column names from and variable names for this. Our y is a function of a constant, that's what this one is, plus the ev1.vec, ev2vec, and ev3vec, and these are just the column names that you have in here. We also specify that we have random effects for all of these things. So random effects means that for the mean and for each one of these explanatory variables, we expect that our estimates are going to differ between subjects. And this is what we are specifying. If we don't specify that, and you feel free to play around with that, you're not going to get as accurate an estimate because that error is going to be sucked into each of the individual estimates, and it's going to throw that off. And remember, we won't be able to generalize then from our population of 10 subjects to the world, right? So the concept is that we would like to be able to generalize because these subjects, even though they're simulated, they're not representative of all the subjects that we could possibly have. And this modeling, this variation is what gets you there, essentially. Once you've done that, you get something that looks like this, and this is just the output of R. And this tells you what you've done, and it gives you your fixed effects estimates. So these are going to be what you're going to be primarily concerned with at the end of your analysis, is the contrasts between these estimates, right? So we don't actually care whether this EV1 or EV2 or EV3 is greater than 0 or not, right? That's what this is. This is telling you, are these significantly different from 0? And if so, what are their value? What we care about is actually going to be the difference between EV1 and fixation, which is the intercept, or EV2 and EV1, right? And that's what the contrasts are that we create, right? So the significance of these just tells you, are these different from 0? And yes, they are. So we simulated our data. I did not talk about autocorrelation with fMRI because that's another very, very big thing that has to be handled. How do packages that you use typically handle autocorrelation? Yeah.
I think FSL computes the autocorrelation on the raw data and then applies it to the regressor, say?
I think it uses a pre-whitening technique, right? So it's basically modifying the design matrix to account for the autocorrelation that it observes, right? I know what FSL does. I don't know. I asked a question, I threw it out there, but I don't actually know what the other packages do.
SPM does take care of it too.
But you just don't know how specifically? Okay. So it's a little bit difficult to take raw fMRI data. We've not come up with a very good method yet of computing the right level of autocorrelation and extracting that from the fMRI data yet. That is a project. It can be done, and so there's no barrier to it, but I actually found that it is easier to account for the autocorrelation sometimes in your model. So if you're trying to model raw fMRI data, and there's really no particular reason why you would want to do this, although I'll go into some examples of why you might, you can do this by estimating or by including an autocorrelation function in your model. So here, what this does is, and I think it's wrapped around here, but you can see that you can account for correlation structure of autoregressive level 1 and give it a form and say that that's within subject, right? So because you're not obviously expecting autocorrelation as you cross over between subjects. And then you can even use an ANOVA to test whether your autocorrelation estimate is better or worse, right? So this is certainly useful if for some reason you have some sort of new autocorrelation function. You think that there might be greater or less than levels of autocorrelation in fMRI data that you've acquired using some new technique or something like that. That's fine. And there have been papers published on what the right level of autocorrelation is in your data and the best way to pre-whiten to get the most accurate results. So that clearly falls within the scope of things that are methods appropriate. But here, in this particular case, since we did not model our data with any autocorrelation, you wouldn't expect that including it in the model would be any better. So it doesn't help. And you find that the first model without autocorrelation is better. Okay, so what we can do now is we can look at the fixed effects and see what they are. And you see that they are close to what we specified, but not a lot. And that's because our sample size is so small. With 10 people and variation around the betas, you're obviously going to have some differences in the estimates that you get. This happens in your own data sets too, right? So you know, if you have a sample, you're not going to expect that you're going to have a very accurate estimate of any particular effect. And you can kind of see this and simulate it here and see what you would get in order to, what you would need in order to get the effect size that you expect. We can look at the random effects, and we can see how those compare to what we put in there as well, and we'll see that those are even a little bit more off, right? Because the standard deviations are going to be less accurate than the actual estimates. So before I go into testing a hypothesis, I just wanted to elaborate a little bit on why one might want to model the raw fMRI data themselves, besides testing some sort of specific hypothesis about autocorrelation structure or something else. We did this primarily at first as a didactic kind of thing, right? To understand what specifically was going on in the fMRI software that we're running, because a lot of these things, like I said, seem to be pretty hidden. And it's much easier, I think, to explain what's going on if you can see it written out in R. And then last year at OHBM, I was at Tal Yarkoni's keynote where he was talking about the fact that when we model task fMRI, we assume our stimuli are fixed effects. So I don't know if people now have heard of this talk or whether he maybe gave another version of it last week or something, but he talked about how if we are studying, for example, the effects of emotional salience on, and we show people happy pandas and sad pandas or something like that, you know, we could look at the differences between those, but not all pandas are created equal. And we shouldn't expect that each person is going to have the same fixed response to those different stimuli as well. So here, our EV is calculated as a fixed effect But if it were something a little bit more complicated, if the design was more complicated and we had a series of stimuli that we showed to people, and they were different images, for example, of nature, of pandas, of whatever, different emotions within a category, we might want to model the individual variants around those and then obtain a better estimate of the group differences and be able to generalize more specifically to stimuli of a particular category, right? So just like people, if we're trying to generalize across a group of people, and we have to account for their individual variations, same thing with stimuli. And that was what he was saying. And he was saying, oh gosh, there's nobody in the room who could do this. And I'm like, hi, so we can do this. We can, you all can do this. This is not hard to do. There's a lure that I'm trying to explore, which is that people don't do this because it's computationally unstable. And I talked to Gang Chen when he was out here for an AFNI bootcamp, and he's a statistician, works with AFNI, and he said, no, I've never had that happen. And I was like, I've never had that happen either. So something to answer, an unknown question. I don't know. He said, like, Tom Nichols says that. And I was like, I don't think it's true. Maybe it's not true. Okay, no offense to anybody who's watching this on video. Okay, so, so I don't know if it's true. It might be. It's something to look into. Okay, so testing a hypothesis for a specific contrast. So now I'm just going to get into this bit about the fact that, as I said, we are not looking at specifically whether the value of the EV beta coefficient is different from 0 or not. What we care about is whether it's different from a different EV, and that's the contrast. And so you specify these things in your software packages by putting 1s and 0s in different columns, but in R you do it a little bit differently. So there's two ways of doing this, and this confused me a lot when I was learning fMRI, so I'm just going to go over this, and I actually at the same time when I developed these notes I had a discussion with people to go over it. Mathematically, you could see how you can actually adjust the EVs. If you want to look at the contrast, for example, between, let's see, this is EV adjusted. If you want to see whether the, sorry, let me just look and see so I don't misspeak. If we want to know whether EV2 is greater than EV1, what we can do is we can actually mathematically rearrange these. So here we create an EV1 that's adjusted, and it's the sum of the EV1 plus 2. And then we have EV2, which is adjusted, and that's just the same as EV2. So basically then the significance of the adjusted fixed effect for EV2 now becomes, because we've mathematically arranged it, the same as that contrast. This is confusing even to explain, right? So the last thing that you want to do is you want to be you don't want to have to mathematically readjust your explanatory variables to compute the contrasts of interest every time you want to do that. And we don't do that. So what we do in fMRI analysis is we specify the explanatory variables in the most solid, straightforward way that makes sense to do, and then we specify what our contrasts are separately so that it makes sense. So this is just for the record, you can do this, but don't. So here, this looks a lot more familiar, I think, to people, seeing the contrast specified here as your typical 0, -1, 1, 0. These correspond to the fixed, for the intercept, EV1, EV2, EV3. We do the ANOVA, and we specify our contrast vector here, I'm sorry. And then we have to adjust these coefficients by normalizing them by the square root of the variance of this combination. And then we can compute a p-value. So here, there's a little trick here, this out business, this ANOVA. What this is doing really is just giving us a calculation of the degrees of freedom, which is a little bit hard to compute. So we're just using a hack in R to do that. And we can see that we get the same t-statistic and p-value for the contrast using this more direct way as we did with the the algebraic manipulations shown above. So this is how you can think about modeling fMRI data directly in R. So I think let us do just a tiny break, and I'm going to introduce now NeuroPointillist, which is some software that is probably of much more use to you, because what we're trying to do there is look at the first level results and model those using a much broader suite of tools than just a mixed effects model. So I'm trying to capitalize on the fact that first level fMRI software does a fine job, but that it lacks a certain degree of flexibility in modeling. And so if you are interested, while we switch gears here, take this moment. Sorry, let me get this slide. So if you go to IBIC's GitHub NeuroPointillist, you can get as far, if you have R installed, and there's a bunch of things to install there, so it's not necessarily trivial. But if you want to, you can take a look there and you can install the packages that are necessary. You don't need them installed to see the demo, but if you'd like to do that, take a moment while I reset up, and then you can get that down. And if you have any problems and you're interested, I can certainly help you with that after. How many people do any sort of work with development or longitudinal data? Okay. Any other people who do work with repeated measures of, okay, other circumstances? What about like genetic groups kinds of things where you have group data? Okay. So this is probably particularly relevant to you because you probably hit walls with some of the existing software that it's difficult to do things. Okay. So switching gears, all of the previous things I talked about was motivated by the fact that we wanted to do this. We wanted to model longitudinal change in fMRI data. And that led to this sort of, wait a second, how do you do this in R? What are the current models doing? What are their limitations? And we had a review paper out on that where we described the software that we've written to do things more flexibly in R. But basically the thing is that if you are trying to measure longitudinal change, you need a bunch of things. You need longitudinal data, you need time measurement, you need enough occasions to measure the correct form of growth. And I think that this is always very interesting. The fact is that you actually need 3 occasions of measurement to accurately look at linear growth. And the reason for that is that if you only have 2, you confound your measurement error with your individual change. And that becomes impossible to untangle. But the most important thing is that you need the right statistical models. So this is just an example showing what I mean by different forms of growth. You can have linear growth, quadratic change, piecewise, all sorts of forms of change that you're looking at in fMRI data, and you'd like to be able to model that. So there are a variety of questions that you would like to ask about change, and only some of them can be currently answered by software that we have available to us. So we can think about how change differs across people, what the relationship is between change and some sort of time invariant predictors. But we might also want to look at what is the relationship between change in an outcome and change in a predictor. So for example, one of the things that I'm looking at a lot is change in cognition associated with change in, say, functional connectivity within some particular set of networks across time as people age with a neurodegenerative disease or not. Along those lines, we might want to ask, if I have a group of people are there different patterns of latent change? So latent is something you don't measure exactly, you don't measure specifically, and you might want to see that, you might want to know whether there are different trajectories of change for different groups of people based on what you can observe. And then you might want to know whether the trajectory of change actually predicts outcomes. So this is just filling in what some of these questions might look like. And we can do pretty well. We can do like the first 2 questions, but the last 3, we don't really have the frameworks to do that implemented in current fMRI software. So there are different statistical frameworks that do allow us to answer these things, and I talked about mixed effects modeling or generalized mixed linear modeling, hierarchical growth modeling, multi-level models. There are lots and lots of names for the same class of models, basically. And those things, that's what I showed you. We got that down pretty well. But there is another class of models, which is latent growth curve models, which is it's a superset of what is what multi-level models encompass, and it allows you to answer a lot more types of questions. So the basic ideas behind this are that you have latent variables. So you observe things that are your indicators. So your indicators may be things like functional connectivity measurements within a specific network at each time point. But you have a construct, which is a latent variable, like maybe a factor. If you're familiar with factor analysis, factors are typically latent variables. And that's something that you don't measure directly. But what that allows you to do is that you can say, okay, here's this change trajectory, which you don't measure directly. You measure some indicator that changes, and you look at that slope or that curve, and then you can look at that in relationship to other unmeasured trajectories. So neurodegenerative process or something like that is a latent construct potentially. Or cognition, cognitive ability. It's not something you actually measure, it's something that you have test scores, and then you know what latent factor might be reflected by those. So you can use these things as predictors, you can use them as outcomes, you can use them in all different ways. So this basically gives you a very, very powerful modeling framework. And people who come from behavioral psychology probably know this very well, have learned this, or at least had a class about it, because it's a very common thing in psychology, but less so in fMRI. And so like I said, these questions that I've highlighted in red have to be answered using a different framework than mixed effects types of models. So the state of the art in fMRI analysis software is, I think, at this point, really AFNI. And this is because what they have done is they have implemented wrappers around the R-NLME, and I believe LMER library as well, to support the full range of multi-level modeling. So even things like comparing models using Bayesian information coefficients or likelihood ratios and things like that, that's not something that you find in those other packages. And so by wrapping R, they've allowed you to do that. And so if you're doing advanced longitudinal modeling, I would definitely look at the packages that have come out from AFNI. But the motivating factor for us was, well, maybe we can just kind of do some of this ourselves. Because we know R, we're familiar with modeling in R. R also has support for structural equation modeling in a variety of packages, Livan being perhaps one of the most popular. So if you can take your fMRI data, or at the very least, and mostly what we have been doing is not the raw fMRI data, we've been taking the statistics from the first level estimates. So the contrasts that come out of our first level models go in then to a longitudinal structural equation model or other model of whatever sort we like, and then we run that on it, and this is what I will show you. So this is just the big level picture from a grant proposal where we have our repeated measures fMRI scans. This is really designed specifically at first for longitudinal, but then we started realizing that other people had problems that they couldn't handle as well. So it's more general than just longitudinal data. And you do the pre-processing and single subject analysis using whatever tool you'll feel comfortable with. We've been using FSL and AFNI. And then essentially what you have is a voxel-wise problem. So it's very easy to parallelize that. It's a massively parallel computation. You split up the voxels into little chunks, and then you farm them out using whatever platform you have. So we have 2 platforms that we are thinking about a lot. The first is just sort of a local multi-core cluster. We can do either a shared memory cluster, or we can do, with SGE, we can do a grid engine type of thing. I think people, do other people use Torque or, what clustering, not clustering, but like what batch systems are people used to using? Condor, okay. So Condor would work, presumably. Anything that can submit a shell script would be fine. And the other option to do that I'm interested in as well is making this a little bit more automatic using AWS. And I think here the question people worry about is money. I can tell you that it costs absolutely nothing to do it for a large-scale study I mean, it depends on your model, I suppose, but if you're doing a lot of like bootstrapping or something like that within a voxel, so you're taking minutes and minutes and you've got thousands and thousands, 20,000 voxels, it might take a little bit of money, but I think that in general I'm trying to benchmark the costs, and it's not very high, especially not with spot pricing, because these things are completely uninterruptible and can be restarted and there's not an issue. So that's something I'm trying to get to, is exactly what those numbers look like for cost. But basically, this is a schematic of what the software is. And there's nothing to it at all. It's really an idea more than anything. So what you do, and I will show you this with an actual example, is you take your data sets, and these are either subject, here I've written different subjects at different time points. Like I said, we've relaxed this idea of time, so it doesn't matter. You just have basically a file with a number of subjects in it with their data. The data could be either the 4-dimensional fMRI data, or it could be t-statistics from some contrast, or a set of contrasts, doesn't matter. So you just, as long as you organize it in some way that makes sense to you, you're going to be the one to read this back later in R, and so you can do whatever you like. So that's where these set labels come. You then have to associate some set of covariates and labels with these things as well. So here it's just got the subject identifier, the time point, the age as a covariate here, and whatever else you like as well. So this data structure you kind of choose by setting up these files, and you're basically just getting the data into NeuroPoint, or Endpoint as we started calling it, or NP as they started calling it in Harvard. So anyway, that's the deal. You don't want to do this for every voxel that in the whole cube of the space, so you need a mask. So you want to limit it a little bit, right, so that you are not wasting time on voxels that don't matter to you. So here, this is just a brain mask. So what the software does is it reads this stuff in efficiently. So I mean, and that's one little trick, is that it tries to be as memory efficient as possible. It creates a voxel data structure, and it creates the design matrix structure. And then what you provide is you provide this process voxel function in a piece of code that is called something like model.r. And so what this does is this just loads that library that we're so familiar with, NLME, and it runs this model using V is your voxel, so that's your brain basically. I actually skipped a step. Often I think it comes from a different matrix, so there should probably be one line in here, but I'll go over that. And so your brain is a function of time, age, and the interaction of the two. And you can do whatever code you like in there, anything. So you can restructure this. You can your data set. You know what we recently did, which I think was absolutely cool, is we in fact put the brain signal on the opposite side of the equation and used it as an independent variable, which I just can't get over. It blows my mind that we could just do that. And so it's absolutely amazing to test some hypotheses about the effect of brain activity on external variables of behavior. So then what you do here is this will be run on every single voxel, split up after splitting up the data set into some number of chunks. And you have a bunch of options here. Usually we like writing out the data in some way so that you can debug things. you can run in parallel if you have a multi-core machine. That's pretty straightforward. I don't like running in multi-core that way because I think that there's bugs, and sometimes the numerical libraries and things take longer than they should, and performance is not as predictable as it should be, and errors are not handled as well. I might be hallucinating, but I just feel that way. So I tend to like to always split it up to actual manual chunks that can be then run on a cluster using the grid engine or something of that sort. And you have two options for doing that. There's a run me script of some sort, and I'm trying to make one for each of the different batch systems, so you just can do it with whatever cluster you've got. Or you can use make to run on multiple cores. And that also, the make file is what's called to put everything together. So it just assembles everything. So there's nothing magical to here. It's just a little bit of an infrastructure for splitting things up and putting them back together, which is often the hard part. So there is an example in Livan in this data set of I believe a parallel process growth model, but just to give you a sense of what this looks like, and I won't go into detail on any sort of models. This latent growth model is equivalent fully to a mixed effects model, and basically this is what they look like. This is the way you specify things. You have 4 occasions of measurement here: baseline, 1 year, 2 years, 3 years. You specify the weights, the regression weights, in order to calculate a slope, and an estimated intercept where the weights are just 1. And so this allows you to estimate a slope and an intercept from longitudinal data in a mixed effects, in a latent growth model framework. But where this becomes powerful is that you can do things like this, where you can consider growth curves in relationship to each other. So this is something taken from this brain injury article, where we're just looking at symptoms. So this is not actually using fMRI or anything like that, but you could. Here, they're interested in the parallel growth of depressive symptoms with post-concussive symptoms. And so what you're doing is you're taking these depressive symptoms and calculating a slope and an intercept for those and seeing how these are related. Are the slopes correlated? Is the intercept predicting the slope of post-concussive symptoms? How are these things related? And by looking at the significance of these regression lines, of these weights, you can determine what the relationships are between these two co-evolving trajectories. And that's very, very important to people looking at a variety of things. It's just not something that we typically do with the brain at the fMRI level. And this is something that I was really excited about. This was a poster that we did. We just looked at the ADNI data and looked at trajectories of functional connectivity in resting state from the posterior cingulate cortex in 3 groups: normal aging, mild cognitive impairment, and in Alzheimer's disease. And what is very interesting to me here is that in the mild cognitive impairment in the AD situation, what we see is that there's this increased connectivity, as shown by the red, to the default mode network PCC area, and decreased connectivity to medial frontal wall you can't see it as well here, but in all 3 conditions. So it seems to me that that this anti-correlation we talk about with attention networks and default mode networks might just be more related to AD pathology. But what's the most important and most amazing thing about this is that this was done by my undergraduate student in a day. So after the data had all been pre-processed. So she knew R from having worked with it on behavioral analyses. So it was absolutely no problem for her to just put this right into NeuroPoint and to do this analysis. And I mean, of course, interpreting what the results meant was harder for her because she'd never seen things like this before. But I thought that having this background in R and being able to take this to fMRI is very, very powerful. And it's a very nice translational way to work with people who come from other disciplines where they use R for structural equation modeling or other such things and communicate with them. So that became a sort of realization that was very powerful. Okay, so I will demo for you and walk you through at least the NeuroPoint software with the time that we have remaining. Okay, so you do not need to have downloaded or installed it. I will show it to you. I will probably make this a little bit bigger so you can see, perhaps. So there's extensive documentation on this on the web and installation instructions. So if you would like to use it, hopefully this will get you going. So when you pull the GitHub repository, this is what you get. So probably the first example to look at is example.raw fMRI. And this is some simulated data that we've done that's very much like the example with the NBAC. It's just sort of a we used a different simulator to do this. So the first thing to look at is the file names and the set labels files. So set file names, there's 2 time points, so there's 2 sets of files. So this just gives you the relative paths here to the different subjects, here S1 through S8, and their time point files. So this is just simulated 4-dimensional fMRI data. The same thing for set file names 2. It's just the same, it's just time 2. So corresponding to that, you need to set up some set labels. So here you notice that there's a lot more rows than 1 per file. This is because this is 4-dimensional fMRI data, so you need 1 row per TR. So there's 2 conditions here, high and low. There is the TR number, and then the subject ID, and then the time. So here is your high, your low. These are convolved. Subject, or I'm sorry, TR is next. So you see it was 1, 2, 3, 4, 5, 6, and so on down. The subject is 1 for as far as we can see here, and the time point is 1 for as far as we can see. So we need to just make sure that this organization, we should have all time point 1 first, and then go subject 1, 2, 3, 4 to correspond to the data that we have. So look, it does seem correct. Okay, so we have here now time point 2, but still subject 1. So that's all there is for getting the data into shape.
Sorry, can you explain again what the high and low columns are?
It's just a simulated experiment. So those are your 2 explanatory variables. They've just been convolved. So exactly like we had EV1, EV2 with the NBAC task. This is some random, like high and low stimulus or something like low conditions. So it doesn't even mean anything really. It's just names for explanatory variables.
I don't think, I'm still not following. What are the values like represent?
So you have some sort of stimulus in a block design, and it's either on or it's off, and then you convolve those with the hemodynamic response. So that's all that is. It's just the task design for the simulated experiment. So just like we saw for EV1, EV2 in the fMRI thing, it's the same idea. So you normally, this is an intense example. You would not normally be doing this. You would normally be doing what I'll show you next. But I'm just showing you the 4-dimensional fMRI because it segues well. And it shows you how it works, because I can run this with everything. I have the data that I need in order to run this. So if you have put the commands in your path, and I have not at this moment, so I'm just going to set my path to include, if you download it, this is what you get. Endpoint and endpoint run, endpoint merge are the things that you care about. So so I'm just going to put those in my path. Make sure that it's there. Okay, good. So now if I go into that example, oh, there's one more thing I need to show you. There's this file called readargs.r. What this is just it's like a configuration file, but it's really just the R code to read in the arguments. I got so tired of typing all the arguments. So all this is the arguments that are going to be used by NeuroPoint. So you can specify all these on the command line, or you could write a bash script to specify them all, or you could just put them in a file called readargs.org, and if it finds them, it will just use them. So I have such sympathy for the programmers who get lazy and just do something easy, and here we are. So this is it. So this is your mask. This gives you, set 1 gives you your set file names 1, set 2, set file names 2, labels 1 and 2. And I should also emphasize there's no reason that they have to be split into time 1, time 2. You can put them all together. And I ended up just doing that later. It was just the way we thought about things first. And then here you have to specify your model, which is the code that's going to be run. You give an output directory, which I'm saying should be sgdata.sim. That's a prefix, so that's used for all your files to kind of keep things organized. This is where your debug data goes. So this is important because as you're developing this model, what you want to do is you want to use the fMRI data to actually test it. And so there are some functions there, and there's a description, a vignette on the documentation for how you can generate this in a file of data and then load that in and then run your model and find which voxel is what. So there's a helpful little command to just take image coordinates to the condensed format that this is in. And then here, this just tells you for SGE how many jobs to put. So really it's SGEN, but it's just how many jobs you want to break it up to. And that really is just dependent on how long you think it's going to take and what your parallelism level is on your computer. Okay, so if you do that, I guess I should also show you the fMRI model. And then this is what we're actually going to run. So this is a process voxel. It's the extended version of what you saw before. The only difference, so you take your V, which is actually a voxel number. So you obtain this from a predefined matrix called voxeldat. That's set up for you by the program. So here, this ugly stuff, E, where you're trying this and stuff, and you give a whole bunch of options, all of this is necessary because when you're running things in parallel, if one of them breaks, then what happens is often they all crump, and then you're left with nothing. So you have to trap errors, typically in a much more sane way, to make sure that you get as much out of it as you possibly can. So in this case, if you error out with one voxel, all the other voxels may be totally fine. You just want to trap that particular error. So that's what this does, is this E business looks to see whether there's any sort of error that you got. And if the model is not null, then that's great. Then you take your contrasts, you look at your output, you compute your t-statistics, and this is exactly like we did in the other example here. Otherwise, I'm liking 999 as a value that I won't see in a t-statistic, so I'm just using that as my error output. And then what happens here is you give names to these return values. The names of what you return is what is used to create your output files in the end. So you can put anything you want. You can return any values that you want from this function as long as they're the same number of things every single time and that you give them names. So, and this is so confusing because you have this incredible amount of flexibility. You can do anything. But probably it's easiest to start with something that's familiar, and this is a familiar case. This is just a contrast. So, the other thing we realized too is a real big pain. If you're returning a lot of things, it's a really big pain to keep in track with how many things you're returning and what their names are, and that their names are in the right order, and that your errors are trapped and stuff like that. So I've been writing generators for this stuff. So you just don't need to do that anymore. You can just generate the code as long as it's pretty straightforward. Okay, so once you've got that, if you say endpoint, it will read in those files, and it will generate the stuff that is in SGE data, which you saw that I already did this before because I was testing it before I came here to just make sure it still worked. But we can just do it again. It doesn't take that long. And then we can look and see what is in the SGE data directory.
Can I ask a question while it's running? Is there any particular reason not to set those 999s to something like not a number instead?
No, it's just if you set it to not a number, it's unclear what it's going to look like from your FSL viewer or your image viewer software. So you might want to set it to something that is mathematically filterable. Because the next time you're going to see those numbers is when you look at your results. Yeah. So it really depends on your viewer and your software pipeline from there, I would say. What is? So this is almost done. we can see here, so since I ran it twice, it's generated things with the hyphens at the end. So I can just remove those. But this is basically what you get when it's done. So all of the, what this is each of those nifty files that you see here. Okay. Okay. So each of these nifty files that you see here is a little bit of the brain mask. And so this is what tells you spatially where this data, the RDS data, actually is. So these are the chunks. The RDS chunks are your actual data in a vectorized format that you're going to actually analyze. And then these tell you spatially where these map to. So, and we know we know how the data are organized, so we know where that goes. This is a copy of your model, because it's really nice to keep all this stuff together so you remember what you did. It's a copy of readargs, so you know what you ran. If you didn't do it, if you did it with command lines, it would just save it as a readargs file for you. And then you have commands runme local and runme.sge. The other thing that is probably important is this debug our data. This is what you would use. You've decided to write this out by calling the specific arguments. This is what you would read in if you want to look to see what was going wrong. You found, say, a 999 at some particular voxel in your data, and you're wondering what happened to the model there, what error did I get? Because it's not saving much about the error, although you could if you wanted to. But that would be what you'd use. So runme.local calls make, which uses the make file to run the jobs on whatever number of cores you have. And that's what the J does. For those of you who are not familiar with make, you can just ignore this. It's a generated file, and you don't really need to worry about it. But all it is doing is it is using the names of the files that you want to generate from the names that it got from the process voxel routine. So it's just saying, here's what I need to do to put everything together. So it's just assembling that. The runme SGE will just submit this to the grid engine and wait until it's done. That's what the sync flag is, and then assemble all the data for you. So this is really, if you have a batch system that you're interested in using that is not the grid engine, it should be a very easy fix to convert that. Because instead of qsub, you would just use whatever script submission thing that you have. You might need to modify the script, the SGE job.bash script. This has SGE submission options, and this would look different for like PBS torque or something of that sort. That's the only other batch submission system that I have a lot of experience with. But essentially, that's what you would do. So that's that. Let me show you one more thing before we adjourn. And that would be a more realistic example, but I don't have the data for it because this is not my data. So this is example.flournoy, and this is a longitudinal data set. So this is described. It shows you what you can do. And there's a few models that you might do. But basically, this has an experimental design with a domain and a target, and it's testing for an interaction of them. And so what you can do is you can have something very simple like this model where you're looking at the effective time. And here we're not doing a contrast, so this is a little bit unrealistic. This is more of an example that just to show how you might read in and process contrasts from a lower level analysis. But here, the key thing is your set file names are different contrasts. So here, and this just comes from what they did, this is a lot more like what probably some of you have from your fMRI analyses. So we have different time points, different subjects, and here 4 contrasts, 1, 2, and 3, 4, which are different combinations of this domain I forget the other contrast. So, but they mean specific things. So what you would be doing then is you would read in some specific contrast and you would do some computation on that and compare maybe the longitudinal forms of change or test some hypothesis about an interaction or something of that sort using data that had been already processed at the first level with your favorite software package. And as you can see here, it's very flexible with respect to what you put into these particular lists of files. It just will simply assemble it into a structure for you, and then you can use the design covariates that you've put in, ideally pick out the contrasts that you want or compare different contrasts, and then from the first level, and then model that in some higher level way. So a bunch of people have been using this to great success, and so I recommend that if you have, especially if you have something that is more complicated that you're trying to do with fMRI, it's worth looking into. I guess the very final note that I want to say is none of this is none of this is doing any pre-processing, and none of this is doing any correction for multiple comparisons. So those are your responsibilities. So the pre-processing, I recommend, as of late, we've been using afniproc.py, and we really, really, really like it. Afni is really awesome. So that will pre-process the data for you and get it into shape for this type of analysis. Now, for correction for multiple comparisons, I expect that probably within the next 2 weeks or so, there'll be some more information added to the website about exactly how to do that. But the basic thing that you do is you estimate from your first level analysis the smoothness of your data from the residuals. And so you obtain an idea of what the smoothing factor is. And then what you can do is you can use, again, AFNI tools for estimating for cluster simulation, and put in that amount of smoothness, use a reasonably stringent P threshold and a appropriate cluster threshold, and then you can calculate what the size of a cluster should be. And then hopefully we'll have a little script for you that it would be very straightforward for you to just run that and cluster correct your data. And this is approved. We know that this is reasonable. At its worst case, it might be a little too conservative, but as long as you do the estimation of the smoothing correctly, that should be pretty good for fMRI data. Now, for other types of data, like if you want to, there's no reason you can't do this, for example, with longitudinal TBSS data or something like that. So there, however, I know less about the error structure of that data and how to appropriately correct for it. So I don't know statistically enough about how best to do that. But conceptually, there is no reason that you need to be limited to fMRI data. You can do this with any sort of neuroimaging data that you can pack into nifty file format. The other thing is that we're also working on more general formats for SIFT data, for Human Connectome Project type of data, which I think would be very useful for some of the projects that have like family structure, and that you want to account for shared variance within pairs of people. And then there is more general, hopefully just CSV formats, so that if you've got some crazy thing that you want to do, you could probably just do it as well, like EEG data or different sensors or something. So it's just an idea more than a software package, but it does kind of work. So I really encourage you that if you have experimentation to do, to try that. Okay, so I think we're almost to coffee break. So if there's any questions, we can do that. Mm-hmm.
So did you just say the input should be at the first level, at the single run level?
You can do either. You can do raw fMRI, or you can do first level analysis level. I suggest first level analysis because I think that probably depending on what you're trying to do, you probably could accomplish it a little bit easier by running your subjects through first level analysis and then using those estimates to bring up to the second level. Now, what you're losing is you're losing the variability, some of the variability around that each subject's estimate. So you better be getting something out of that in terms of putting that into a more sophisticated model than you could do otherwise, right?
So is it possible to use the second level individual maps? Because I know with FSL, I think for event-related designs, there's some question about how the scaling works at the first level with the single lens. that makes those estimates like less reliable, or it's unclear how they're scaled exactly?
Well, we've been using t-statistics, so those are scaled, and so you can use those. It's true that if there's benefits to using the estimates versus the statistics. Statistics may not be as good as the estimates, but you can. I think that in practice you can certainly use either. But like I said, you know, that would be something to do if you can't do that. I think in your traditional frameworks, because you're trying to just go beyond that. So I would definitely think that, you know, if something has been implemented well, say by the AFNI folks, it would be a good idea to try that first, because they probably have accounted for the statistics more accurately than I would be doing, certainly. But that said, there's a bunch of things we can't run. So as you're you know, moving ahead with more advanced models, I would imagine that this will develop. My hope is that it would develop. So practically, if you can do this stuff in R, and I've even had colleagues who are writing me that they're trying to pass up the variance estimates as well. And there's no reason you can't do that either. Just plop that into your list of files and you can do that and run a Bayesian inference on that. So if you're comfortable with that, no reason not to. The question will ultimately be just, you know, how long does it take to do that versus how long does it take to wait for somebody to either write you the software or write it yourself in a fast enough infrastructure. And you know, I feel like computers are pretty cheap. I feel like my time is pretty stressed out, and I feel like I think that we're hitting that sweet spot where we could start investigate some of the assumptions about how long things take and how expensive they are in the cloud. So that's really what drives this. Is I think there's a change happening. Any other questions? Just looking uncertain. Okay. All right, then. Well, thank you very much. And I'd be more than happy to answer emails or questions if you do try this afterwards and you want to know something. But otherwise, I think I'm the only person standing between you and coffee. So I want some coffee, too. So, thank you.